Well, Tony had set this up with everyone, so I think we're just really going to cover getting acquainted with it overall. Do you want me to do our kind of normal intro with the slideshow to start, or if I didn't know if you had something else that you wanted to start with first, Tony?
Um, getting the ExoLab ready. What do they need to do to get started on the 24th? With the
Okay.
ExoLab? Yeah.
Okay. So let me bring.
Can I just set a little context here?
Sure.
All right. So as you guys probably know, we lifted off out of Wallops, Virginia on Saturday. And early Monday morning, we got grabbed by the Canada Arm and we're now berthed on station. I understand they're unloading us kind of as we speak. They'll take our lab and slide it into the rack and boot us up. We're going to let that run and go do some diagnostics for the next 48 hours or so. But we're planning on starting schools as early as the midweek here, Wednesday, the 24th. You don't need to start on Wednesday. You can actually wait a few days. As soon as you do start, you'll be synchronized to the very first day. So if we get it booted up tonight, for example, when you turn your lab on, you'll be looking at day 1, minute 1, just a few days prior. We do that because we don't always get a daily download from the National Lab, from the space station, and we just don't want a gap in your data. So we have that little buffer in there that allows you to have a continuous experience for your students. So I just want to share, that's pretty exciting to see that happen. And of course, what really excites me is just the name of the Cygnus spacecraft that went up there. It's the SS Katherine Johnson. And of course, we're here just rounding out Black History Month. And she's taken us right to the space station here for Women's History Month. So what a beautiful bridge to be able to discuss both those things. One thing that really inspires me with this program is all of you guys. We really kind of look at this in a very interdisciplinary way. So while you might be focused on physics or on biology or chemistry or some focus of engineering, there's the arts, there's history, there's so many other things we can talk about. And if you have questions or you are looking for ways to engage, please reach out to Tony, Lori, or myself. Michael's not here as well, but Michael's our director of ed. Would love to be able to find a way to integrate some thinking that you have. But I'll be quiet now, Lori. I'll let you take the lead.
All right. So I'll share my screen with you. Let me go ahead and give you a preview. Were any of you actually on the launch party on Saturday? Yeah, we had a couple. Wonderful. All right, so want to first give you a look at our Flight Cube Lab. This is it's a 4U unit. It is sealed up because we have bacteria in there with our plants that are nitrogen fixing and friendly for the most part. But the space station, they do not like extra bacteria floating around. Excuse me. So we are sealed up in that box, and Lisa Turney is our ExoLab-8 Astromoji mission specialist. She's a technology teacher out of Linwood, Kansas, who was selected to be our virtual teacher in space. So what you're going to see here with the Flight Cube Lab, it's a little bit different than the classroom or ground stations, in large part because we are going to fix the plants while in orbit. And so that's what this large black box is right there in the middle. It's been 3D printed components, and we have 3 test tubes in the flight unit so that we could fly 9 seeds. And so you'll have 2 test tubes in your ground unit, and you'll put 3 seeds each. And so you'll have 6 for your ground samples. So the seeds are in the same type of agar. You're going to see that they are also in a little bit of biostrate felt, and that is to try to delay germination. When you put clover seeds directly into media, they start germinating in about 12 hours. So we wanted to slow that process down just a little bit while we're in the dark. And then within that black juice box there, we also have a little bit of water that's in the tubes so that when we are ready and powered up, we will activate this system and you can see the little green tubes going down into the test tubes. And so there'll be about a 0.5 milliliter of water that will go onto that biostrate felt that will then wet the seeds and then start our process. So this is definitely a technology demonstration, as well as this is the first time we are trying this technique to avoid germination while in the dark. A remarkable thing happens to clover when you try to grow it in the dark. It actually grows really fast and the stems become very elongated. So we've worked over the last 6 months or so to try to prevent that so that we have healthy plants growing on station. So this is a good shot there of what you're going to see with the flight box. And then up close, we have 2 cameras that will be taking pictures. One in infrared, and then one's kind of a fisheye camera, I believe. And so that'll be the image that you mostly see when we get onto the platform. And then that was our rocket from last weekend. But let me flip over. I just want to hit a few highlights. Ted, I don't know if you want to say anything about, quickly about anything about past missions that we've done.
Yeah, well, I can lead up to that. Tony and I formed Magnitude back in 2013. Took us about 4 years to get our first mission in space. And actually, the 4-year anniversary was this month. SpaceX 10 was the rocket that got us up to the space station on the Dragon. And we launched out of historic pad 39A. The first time that launch pad was used since the end of the shuttle era. So it was really great to see that. And 4 years hence, we've got our 8th mission going up. When we first conceptualized what ExoLab could be, it was really a laboratory outside of Earth, thus EXO, and it was across many different domains of science and research. We rapidly discovered, as we were kind of just going headlong into this, that it was probably ideal to specialize. And so all of our experiments have been plant biologically based. There's obviously, we're flying some bacteria now in the symbiotic investigation. We'll probably kind of stay down that path. But if you have any students, or if you yourself are interested in things like crystallography or material science, microfluidics, once you start understanding the nature of microgravity and how that can affect any of these domains, we're very interested in what you may be thinking up in your mind, what your students are imagining. I'll just plant the seed here, like literally here or figuratively. I am the education chair of the U.S. National Laboratory now. I work with a subcommittee in education. A lot of it's kind of oriented toward universities, but I'm really open to hearing what you guys are interested in doing in space for your students. So we have just a slender part of what we do with Magnitude, but there are many other opportunities that are up there for you if you want to get into other aspects of this stuff as well. So I guess I'll end it with that, rather than say,
great.
Welcome to space.
And so this is our amazing PI team who's really been helping this last year with curriculum development, as well as all of the experimental and ground trials to develop out the protocols. So good international representation. So you'll see some of these names and faces as we do our live sessions and pre-recorded videos throughout the month of March. And then you'll see some of them as the authors of the lessons on the platform. So they are a lot of fun, and definitely if you have that chance to log on when we do the live sessions, please do so and bring your students. Steve likes to wear some fun goggles when he's talking about his lessons and such. So they're all very dynamic educators. Many of us have participated in a lot of different types of project-based learning to bring to students and really integrate space science in different ways, whether it's biology or physics lessons, many different ways that you can approach it with really all different age levels. So through project-based learning here, we aim to teach scientific inquiry by having them grow ground trials and have access to that flight experiment while it's on station. And our science aim is to achieve root nodulation with the symbiotic rhizobia bacteria with those clover plants under the stress of spaceflight. So this is definitely a demonstration mission, and it's never been tried before. And so welcome to the first time of trying this in orbit. It's pretty exciting. So you can look forward to, and have your students throughout the project keep notes and folders and documents throughout their lessons so that they can create poster presentations for the end, even digital ones. Like this is one that my students did the last year through Google Slides, just summarizing everything that they've worked on through this and really represent it out in a very visual way, as well as teach them science communications with writing, data analysis, tables and charts, as well as images and such. So we really want to emphasize this process of scientific inquiry and reiterate to them that we don't have all the answers and that they are going to really contribute to authentic research with these clover plants. Many of the lessons have claim, evidence, and reasoning type questions embedded in them so that they really become those good scientific communicators. Even from the youngest grades, there's several lessons that Lauren produced for early elementary that start introducing this concept of CER. Throughout the platform, you'll see how it's organized with a core sequence of lessons. And in fact, I'm going to skip over. Let me see if I'm going to bring up the platform. I want to show you. I know Tony will do it too. The way our lessons are organized. This is a good place to show that. Let's see. So new lessons that we put up that will have videos in them throughout the mission, they're going to appear up in this top row. So if you missed the launch day party or you missed our flight readiness review panel last Friday, you can go back into those lessons and the videos are embedded into them along with initial information about the mission. And depending on the grade level that you are working with, you look at the top here to see their grade level designations and kind of grade ranges and different options. If you have students working with the optional, that student lab kit, the $10 ticket to space kit, you'll want to direct them this week to start looking at this lesson so that they'll know what to do to prepare their kit when it's time. And yes, I think I star in a video or two in that one. Thankfully, I had an extra pair of hands from my sixth grader to help with that demonstration. But then you'll be able to work through these columns of the different missions. And these are the core sequence lessons that you can work through really in any order. The one that you'll want to really pay attention to this week is the mission 4, because this is where the specific flight protocols are. Now, we didn't, for the elementary and the middle school, we didn't want to make it overwhelming for them. So we gave the simplified version of the protocols. But to set up the ExoLab as teachers, you'll click for teachers and curious students. You'll click here and it'll bring up the full PDF. It's already embedded as a PDF, full screen in the high school lesson, but we thought it would be a little bit too much for the younger students. But this is what you'll want to refer to this week and read through so that you understand how to set up your ExoLab, as Ted said, around February 24th. Do really emphasize sterilizing everything, particularly those test tubes and anything that touches the agar or the seeds needs to be sterilized. You do not want to have any type of overgrowth of mold or fungus, because that will cause stress in the plants and they will not nodulate well at all if you get other microbes mixed in. Okay, so these are pretty simple. You have a picture here of the rack that goes in there. I will say, as you've got these test tubes in the rack, definitely use the caps that came with it. It really helps balance that rack well to stay up. So avoid that tipping hazard. So that's our lesson protocol, and it's in lesson 4 is where you'll want to direct. Also within that lesson is good for your students to start reading through on how to make these observations. What do you wonder? What are you noticing? And then great diagrams here that Michael drew out so they become familiar with the terminology, because when they record their data into the table, they'll have to use these words to describe what they're seeing. And so that you can refer them back here to these different stages. And then, you know, even pigments tell us a story as to what's happening in that plant. So getting used to, okay, is it green? Is it really a solid green, or is it more of a yellow green? Do you see orange in there? Do you see purple in the leaves or the stems? And then how to make the observations, what you're looking for kind of week by week. And then a PDF if you want to have them fill it out, and then it goes kind of week by week. So the actual experiment data, I'm going to switch accounts here. You're going to put into a different section where the experiment is, and I'll bring up one of mine.
While you do it, Lori, if you guys have any questions, we can pause for some questions or feedback.
When should so when I log into the platform right now, it tells me that I don't have access yet. Is that because I haven't received my box yet?
Okay. I think I can fix that right away for you.
Okay.
Yeah, just make sure your license is activated. That's the problem. So
Yeah.
I guess,
Sometime Tony, me here, forget.
Yeah,
don't mind me.
No
Got to
problem.
pull the big lever.
If that's the worst thing that happens today, then I'm good.
right. Yep, Tony will get you fixed up there. I think my pictures are a little slow in loading since I've got so many tabs open here and running Zoom. So hopefully they'll load soon.
I
But
think
when
I think you just have to drag back a little bit.
yeah, I've got the little icon here at the moment for just the image. Okay, so they're there. Oh, here they start coming back. Let's see if I can get the other one to load at the moment. There we go. So you'll see. Oh, I don't think I can expand it very well. Right here at the bottom of this left test tube, you see a little white extension off the root. That's a nodule. It's very interesting with clover of those nodules can form anywhere on the root. It could be right here at the base of the stem, just as the root starts, anywhere on the branch or the long taproot, and this one just happened to form at the very bottom. And then actually in my, on my left-hand side, there is a root that grew very close to the edge of the back of the test tube, and it made the nodule impossible to see from the camera side. So just because we don't see it doesn't mean it's not there. And so we have to sometimes look for some clues with how the plants are doing with their color. And how fast they're growing, even comparing each other. If one starts really taking off and growing a lot faster than the other and is really nice colored green, then it's often a sign that it is getting a little more plant food than the other one. So you'll look for different signs of not only the obvious nodules, but even secondary signs that nodulation could be occurring. So up here in the notes section is where you'll open up and be able to fill out what's happening. Ideally, if you can get observations and data plugged in every other day, a couple times a week, but at least once a week, it would be really helpful. Having a variety of these ground trials so that we can start looking for trends in the data, especially as we get closer to root nodulation. Back in the fall, there were times with our trials that we had nodulation in 8 days after planting. Now, the average tends to run between 18 and 24, but it can happen a lot sooner. And so we want to start looking for, okay, well, we're getting a lot of people reporting nodulation now in these ground trials. What's happening with our flight experiment? Can we compare to that and say we should really be seeing some nodulation by now? The L1, L2, L3, so that's going to be working across your test tube from left to right and keeping those seeds in number kind of consistent order, and using both the quantitative and qualitative description. So again, here describing those shapes, the colors, the structures, and anything that's particularly noteworthy of for each of the plants. Excuse the background noise. My children have decided to eat dinner. All right, so that's some of what you're going to see as we get started with the experiment. And then if you click charts, this is where you're going to see the comparison data come up. Let's see if it loads. There we go. So looking at CO2, humidity, notice there's some good patterns, temperature patterns. Sometimes one will run a little warmer than another one, which is kind of interesting considering these are near each other. Light. So one, if you use a variable of blue light versus red light, we are planning to use magenta in the flight experiment. So you may just want to run both of your tubes on magenta light, though you can change color if you wanted to do a color comparison. And that's what I had been doing in my last ground trial here, with magenta on the right and then blue and red with the light comparison. So flip back over here. So among the different lessons that you're going to see on the platform, a lot of different paths you can take it: plant physiology, legumes and nitrogen cycle, tropisms. We really want kids to understand that we have to really become better at food sustainability on Earth, as well as food crop production in space. Our long duration space missions will require some supplemental plants to provide the astronauts with nutrients, because the prepackaged food will break down. And it's got a great social studies connection with early explorers with who were dying of malnutrition. So the fresh food's important. You can go deep into some of the physiological impacts of space flight and how nutrition plays a role in that. And the quantity, the mass, there's often good classroom challenges of, okay, you have this small size box of things that you can take with you and it can only weigh so much. So what would we take? How do we get the most bang for our buck nutritionally, and with even the type of seed? That's one of the reasons we chose clover seeds, is because they are so small, and that we could get 9 total flight samples up in this ExoLab to have multiple trials at it. So some of the different hardware that's up on station currently with Veggie and Advanced Plant Habitat and our ExoLab Space Tango Cube Lab. And if you've never grown legumes before, this is what we're looking for, is these nodules like you see on the left, and you can see the roots on the right do not have any. So on the left, this is a sign that nitrogen fixation is taking place and they're enjoying a nice symbiotic relationship. So as I mentioned, those nodules can form right at the base of the stem, and it's really messaging that takes place between the bacteria and the clover. The clover have to agree to let the bacteria in through the root hairs, and if the plant is stressed, it will not let them in. And but if they are like, okay, well, this sounds like a good deal, and the bacteria are offering nitrogen in a form the plant can use, and then in exchange, the plant is providing carbohydrates to the bacteria. So you'll see these nodules, they're basically like little infections where the bacteria is growing and replicating. So great opportunity to teach those biogeochemical cycles. If any of you teach upper level grades, higher level students, the nitrogen cycle is one of the most confusing to them, and this will be beneficial. You can also take it the route of teaching them about chemical versus biological fertilizers and the consequences of chemical fertilizers with runoff, eutrophication of lakes and waterways, dead zones, algal blooms like we have here in Florida. So you can take it many paths. Also good time to integrate art. And really, this is where they can pick up on the pigments. What does that plant look like? Not just in a pencil drawing, but add some color to it. And we can often look, like I said before, as to that health of the plant based on its colors. And then integrating that scientific vocabulary by labeling. Tropisms will be an exciting one to look at too, because we find with red clover that they are very sensitive to light, and to get those stems to grow up and those roots to grow down, it likes a light source. So when we power up the experiment for the first time, we'll see if any growth has taken place, and if our little seedlings are confused at all, and what impact it's had on sending them through the G-forces of launching on a rocket and now being in an environment that's microgravity. What's going to happen to that root architecture, the taproot, those lateral roots? How's that going to compare to what we see in our ground trials? So even just looking at and drawing out the shape of the roots, that makes a difference. You can see on the right-hand side in that upper test tube, just under the 48D, yes, that is a root that grew up, and that was one that had been in the dark for 48 hours. Microscopy is going to be the focus of our post-flight science. After we fix the plants in orbit, we have fixative in that juice box, and when we say it's time, Space Tango will send up the signal, and they will release that fixative and freeze the plants in time. And then once we get them back later this spring, we'll focus on microscopy. We'll see the condition of the samples. We might be able to do genetic research, but it's going to depend on how long they are up there, how long it takes to get them back, and the temperature of all during transit. So you can look for lessons on staining and some demonstrations of using the microscope, especially for those who are distance learning still and won't get an opportunity to do the hands-on aspect of microscopes.
Lori? If
So,
I may,
yep.
I noticed Jerry Jonas. Hi, Jerry. This is a recorded session, so welcome. Everything that you weren't able to get a hold of, we'll have a copy for you.
I apologize. We had way more snow than we expected in mid New York State, and I just got in from that.
Well, I'm glad you made it there
Oh.
safely.
Yeah.
Definitely.
I guess one thing I'd also like to note here on this microscopy post mission, we were originally scheduled to come back at the end of June. And through just great fortune, we are coming back early on a historic Crew 1 Dragon return in early May. So if you really want to get your students excited about the future, follow along with what's happening with Crew Dragon and the commercialization of space. They're going to be riding home in a historic mission on the return flight. So it's quite a feat, and we're really excited to be part of that.
Definitely. You can also take this direction with students to look at soils and permeability, porosity, growing clover, because they'll have seeds. If you've got those student kits, you're going to have an abundance of clover seeds. And so even having them go out their back door and grab a handful of dirt and have them grow them and compare how the plants do. Clover is a cover crop and is used to help enrich the soil. Typically in the off season, farmers will use it to get nitrogen back into the soil, and then they'll just till that clover back into the soil and grow their other crops. So when you relate it to the moon and Mars, we know that dead soil on Earth doesn't grow anything, and that we need those organics to and the microorganisms really to make it a productive growing media. And we've got some issues when we look at growing in Martian or lunar regolith. And if you are particularly in an area that has a lot of red type clay, go out and grab it and call it your Martian simulant, because and see how those roots do, especially the clay-sized particles suffocate the roots. And so it turns into a good multidisciplinary lesson when you even expand it in this way. So I'll stop there. This is what some of the platform that we covered, and we're just really excited that all of you have decided to join us on this mission, and I'm really happy to take any questions and help you all along the way. Have any of you gotten started with the lessons on the platform yet in assigning them or working through any of them?
I've showed several in, we're actually in-person teaching in small school. We're getting away with that in the middle of New York State so far. So I've been able to show some live and discuss some of those. That's been good. I've just gotten a notification that after I left today, our ExoLab has arrived, hopefully. So hopefully when I get there tomorrow, it will be there. I've been waiting for it for a long time. So
I know.
Something about the post office box, and that's not usually the way that we get things. So I may still have to track it down.
I see.
Yeah,
And Jerry, actually, over the weekend, we have gotten 2 other teachers from your school asking about this this mission. Do you know anything about that, or do you know if they are colleagues of yours?
2 other teachers from our school asking about it?
Yeah.
I had not heard about that. So
Okay.
we're a small school and we're a Christian school, so we have a small circle prayer meeting with all the teachers every morning. So I'll have to ask and find out.
I'll share the name with you right after
good.
this call. Yeah.
Okay, that would be good.
Okay.
They're going to be quite jealous. You're going to have that ExoLab in your classroom.
Well, I guess there's I know that the Traverse City Libraries there have a little different configuration with people coming into the library now that you're going to be opening up again, or your libraries, and engagement with student kits. I'm curious if anyone wants us to walk through how that might work alongside your ExoLab. Knowing whether you're hybrid or at home or right there in the classroom can also help. I don't know if you would like us to walk through what that might look like.
Lori has a video made for that.
Mm-hmm. I'm just walking through it in discussion since we're here now.
Sure.
Essentially, the little kit that you've received, it's advised, I guess, if the students are of younger age, to work with some parents. But by middle school, I would hope that they know how to boil water, whether it's the microwave or on the stovetop. But it's just really just mixing that powdered agar in that solution. And then, Tony, you have to bring up the actual video. That will basically show your students and you the step-by-step. But I'm assuming everyone's got the student kits as well as the ExoLab. Is that right? And really the only difference between the student kits and the teacher kits are you're getting those borosilicate tubes that fit inside the ExoLab with the rack. and then the forceps to allow you to kind of go down in that tube there and plant. But other than that, the materials should be the same. And if you haven't received a mission patch, the fabric patch, ping us. We've got a few of those left and want to make sure every teacher gets at least one of our mission patches. I've started collecting them from all our missions, and it's nice to be able to add a patch to the collection.
Michael, you should have one for every teacher.
Mm-hmm. Yeah, I have some extra ones here too. So if you haven't received that actual fabric patch, let us know. We'll get one out to you.
And Jerry, you have yours. Well,
I have mine, and to respond to your question, today I was orienting my middle school, and I've got 7th, 8th, and 9th in various classes. So I was orienting them and sending home packets with them. I used to work in research lab and worked with Augur a lot, and a suggestion for your instructions in the future, Augur is very likely to have explosive bubbles if you don't. So I didn't feel like that was emphasized enough in the instructions. So I did a little bit of extra talking about. The second thing is that the Augur powder tends to sink to the bottom even while you're microwaving. So if you're microwaving, it tends to be good to pull it out, stir it a couple of times, and stick it back in.
Yeah, particularly we try to emphasize through this and the videos and the safety notices that they have to be very careful in preparing it, especially with younger kids. So if there is a concern with younger students, then they can look at using an alternative media, whether it's the soil from their backyard or growing them in cotton balls or paper towels or something else. If they don't want to try to mix the powder, we certainly don't want young students to do that unsupervised, because the glass will be very hot. And yes, it can bubble and it can burn as well. So they can't leave it on the stove and walk away from it. But thanks, we will definitely keep emphasizing that safety aspect with it. We do have a video in here as well, that if they're going to upcycle a plastic water bottle, of just how to very simply take one and cut it to serve that purpose. Again, being careful that the plastic, it will be hot as you pour it. That's another reason why in the instructions, after you remove the agar from if you're using a stovetop, or the microwave, let it sit for a few minutes, for about 10 minutes. And then that definitely helps with letting it cool just enough to where it's not, melting plastic from a recycled bottle. You can use glass jars, olive jars work well, jelly jars. But they definitely need to have some of these other supplies on hand so that they can do their student kits. And then we walk through the procedures through it. And letting it cool on that angle is important. We discovered this as a technique that allow the roots to get greater access to air. They tend to nodulate better, and that makes sense since the rhizobia want to take that nitrogen from the air and fix it. So having that little bit of tilt helps with the plants to grow their roots down the angle of the agar, though sometimes they'll go straight down into it. You can't control it all the time. They will have some extra rhizobia powder left over. So if you want to have them save it for another experiment, then you'll be able to do another trial after they do this initial one.
Yeah, I'll just add, our philosophy here is really kind of coming at this from first principles. And the general thesis that Tony and I had when we got started is we wanted to have someone engage with our experiences and evoke some kind of emotion. And it's like art, right? If you look at a piece of art, you might be repulsed by it, or hopefully you're curious or enamored and you want to learn a little bit more. And so something like this extraordinary mission to space, we hope can stoke the coals of curiosity with your students, especially during these times of these kind of weird configurations of the classroom, or what might be happening at home during these times. Fortunately, it seems like we're rolling out of that period, but I think it's a really great opportunity to engage our students in thinking about perhaps a slightly different way of going about the regular routine. And I think to emphasize that, what we're really excited and hopeful for is that your students share some of their work organically or more directed at us. So if they do a TikTok video about the mission, on them, not on us saying, here's a lesson you're going to have to do, or if they write a poem or they do some artwork, we'd encourage you to share it. Lori and our team will be running some live broadcasts starting on Thursday, talking about the summary of the mission up to that point, what we're looking forward to, and a chance to just kind of build community. And then in your mailboxes on Monday morning, you will have a shortened video that's kind of a synopsis of what's occurred the last week, if you want to use that to flip the class. And so having your students' content in there, I think, first of all, there's some pride in your students' work. I think they can get a little more engaged if they would like to contribute in that way. We can't guarantee that we'll be able to cover everyone's work, but we'd love to see it, and we'll do whatever we can to highlight some of the work that's being done out there. So if your students are maybe less excited about science and maybe more excited about media or some other aspect of what we're looking at, by all means, they want to write about, Katherine Johnson as a historic figure, have them write something and we'll promote it. Whatever we do here for this particular mission, we always make sure we roll it up to the national lab so they get a sense of the impact that we're making. Without their support, and they manage half of the upmass that goes to the space station, and they manage half of the astronaut time. Without their support, we wouldn't be able to extend the missions that we're doing out to everyone right now. And so anything your students can do that they're excited about, it's not like an assignment, we welcome that to forward that if you're interested in doing so.
And if they have questions, because we would love to have student representative questions that we can answer through the videos, and say, Timothy from Kansas City wants to know, and then say, oh, well, the same question came up from Zamrith and Sri Lanka, right? And they had the same idea. Let's talk about this. So and to help make those global connections. Sometimes students are afraid to ask questions, like they're the only one thinking about it, but they're really not. And so we want to help them get comfortable with, there's lots of people being curious about this and asking questions. So definitely send those to us by email if you want to have your student kind of showcased in that way too.
There you go. Laura, you kind of some location dropping there in terms of locations around the world. Tony, you want to share about some of the excitement of the global community that we're kind of building? And there's an opportunity, if you want to coordinate that with us, to have your students perhaps engage with students in, like, Nigeria is joining us now at the last minute. We just got word from them this morning, or Kenya or South Africa, or in Germany or Romania, Canada, Japan, Australia. They're just kind of popping up. And so that kind of outreach and engagement, I think it's an interesting thing to consider. As we look at this experiment, that symbiotic relationship between the bacteria and the plant, we hope it inspires the potential of human settlements off-world. The idea of putting a garden on Mars is, I think, fascinates a lot of people, young and old. But probably more importantly, there are a lot of challenges on our planet right now, and this could be a really great investigation to think about what we need to do on the agricultural front. And so your students, again, can kind of take this in so many different directions. We're excited and encouraged by the feedback we get. So please don't hesitate to reach out to us. So yeah, Tony, I don't know if you want to say anything about the
I want to add one more thing. This is a really good opportunity to involve the parents too. Now, because every single student, they have their own account. They should log on together with their parents at home, showing them what they are working on, images and data coming off from International Space Station, over the weekend, over a holiday, and they can log on from anywhere.
Now, I do have a question about the student accounts. Does each student who has a student kit have an account, or is it the students that I designate as part of my class have an account? Does that make sense? Because I ordered like an ExoLab for the classroom and the teacher's kit, and then I also ordered 6 student kits because I'm going to have them in groups because I'm also in person, just like Jerry, where we've been in person since August. Also a small school. That's probably the reason why it's worked so far. Thankfully, haven't had to go into quarantine. So it's been a challenge, obviously, you know, because we wear masks the whole time, in addition to other things. But so I've got 6 student kits because I want them to be in groups and I want them coming up with their own experiment, right? Like, what do you want to do with your seats kind of thing to compare with our ground control. And I even told them to keep a notebook. So they're all getting their own notebook so they can record their data and all of that. And so they're really excited about that. But in terms of like the account, so right now I'm set up as a teacher. So I guess I have the autonomy to go in and set up my classes and then
That's right.
invite my students to my class. Is that how that
That's
works?
right. That's right.
Okay.
Yeah.
Okay.
Yeah,
Once you have a class created,
Okay.
share the class code with them.
Okay.
So for
Yeah.
so for
Lori is,
instance, you'll add a class
Mhm.
and give it a name, and then it'll bring it up here and it'll assign this class code. And so you can then share this link, or you could actually create them because down here you can assign and add students. So you can add it that way too. And then your experiment will be connected here is where you'll connect your ExoLab with this particular class. And then all the lessons that you've assigned will show up here and you'll be able to directly access their notes. It also shows up on your teacher dashboard of your different classes, different lessons that you have. And lesson progress from folks. So essay results, yeah. So
Okay, so then the student kits, those are just kind of for them, like for them to do their own experimentation, but they don't have to, that's not how they get their username. I'm the one that kind of assigns or gives them the link, basically. Okay.
Right, you set them up, you set those accounts up on the platform, and then you can share those, the username and password. I think each student kit, you get one account. So you have to have, I think, 6 different email addresses. Isn't that right, Tony? For the students?
Yes.
Yeah. Okay.
Okay.
Sounds good. That's awesome. Oh, one other, do you guys have specific emails that you want to, because I know, for example, if I have a question or a student question, do you want me sending it to a specific person versus student art and presentations, or do you have just one email where you would like all of that sent to?
Hey, Lori, can you put up your screen again? On the lower right-hand corner. No, on the lower right-hand corner of your account. This is where you can find some of the answer to your questions. And you can type it in, for example, like Wi-Fi connectivities. How do you connect your ExoLab to your school Wi-Fi? One thing to add is that some school district it require the IT department to whitelist our device. And if they need to do that, let us know. We will share the MAC address with your ID department so that they can whitelist it. And also over here, you can send us a support ticket. All you have to do is click right there.
Mm-hmm.
And you can all this email will go to our support, and a number of us will get that to answer questions.
Okay, thank you so much.
And for like sending in the student work, since you'll be doing attachments, the best way to do that is hello@Magnitude.io. You can try to send it directly to one of us. I've got some travel coming up and may have a little bit more limited access on
You have
some
to share what
days.
you're traveling for, Lori. Come on. We're really
So, okay. I am going to the high seas Mars Moon analog on the top of the volcano in Hawaii, and I'm taking an ExoLab with me. And so we are going to have a ground station at 8,200 feet on a volcano for a couple of weeks. So it'll be pretty exciting to participate in a moon analog mission through high seas. And I'm a graduate student with the University of North Dakota as well, studying human factors and actually studying extreme environments in this semester. So my professor is absolutely thrilled, and he's been even following along with our ExoLab mission too. So yeah, so you can look for that in the middle of March, and hopefully we'll be able to send out directions of how to connect in and see the plants growing when I'm there. So, but I'll have a little more limited connectivity.
Oh, that's awesome. Thank
our
you.
own little Mark Watney.
Yeah, so I'll have some videos and things that you'll be able to show your kids. So it'll be exciting.
Can I follow up with a question on the student accounts? I went in and started student accounts using email addresses I had, and then found out that some of them needed to be corrected or deleted. And I can see no way on the platform to change anything once it started. It sounds like they got a notification, but there's no way for me to go in and delete and change.
Yes, send me their names and email addresses to correct, and I will do that for you.
Thank you.
No problem.
All right, we're getting close to the top of the hour. Any other questions in preparation for the start of our mission?
I have one, Ted.
Yes.
And it seems so rudimentary. I can't believe I overlooked it. But I feel like we have the parent-teacher kits and not the student kits and not the teacher kits to work with ours.
Okay, so let's see. Did you run ExoLab-7 with us?
Yeah.
And 6? So okay, so you have the borosilicate tubes then, right?
Yep.
And the rack. So essentially that's what it would be. I think the only thing you're missing then would be the forceps. Okay. Unless you need another kit it's essentially a student kit is what you'd use to set your
okay. Yeah. Just forceps. No, that would be it then. Yeah.
So let's get some forceps out to you.
Okay.
All right, let's do that.
Yeah, it definitely helps to have those longer forceps to get into those tall tubes when you're placing the seeds down in that auger, because you want to like press them in slightly. Otherwise, they like to go wee down the slope of the auger.
How do they go? No. So do we have the individual addresses for each of the libraries? We do, right?
Yeah.
Okay, we don't just send them central.
Or you could send them to me, and that would be fine too.
Yeah, send mine to Andy. This is Rosie over here.
That's easy. I can just put it in one package and I'll
get it. And Ted, while you're send a couple of patches too. I love my patches.
Okay. So. All right. Now, we actually have a limited number of stickers left.
We'll take stickers too.
Yeah. If any of you could you send a note to hello with a quantity request? And
sure.
What I want to do, if you have a smaller group, we can probably fulfill that. But if you have a lot of students, I wouldn't want to send just a few. I'd feel like they might feel like they're disadvantaged, or some are getting it, some aren't, unless you want to use them for reward or what have you. But we'll go through our count. ExoLab-8 is underway, and if I've got some patches, let's get them out to you, right? They don't do anyone any good in my office.
Yeah.
So send us a note, hello, right, at Magnitude.io. That's great.
And Tony has I don't know if you have any of the boarding passes left, but even that's a nice thing to give to students so that they can be part of it. So
I think they all got them, right?
Did all of you get the boarding pass with your kids?
You put those up kind of late there, Tony. So I don't know if everyone got them. I don't know do I have a copy of it?
I won't know until I get my boxes. I'm hoping they'll come late. They'll come soon.
I think Andy got his, Jerry got his, Michael got his.
These are kind of cute, really, in terms of these basically look like an actual ticket maybe perhaps for younger students, but some of these things are pre-selected, but the trip, is it one way round trip or I don't know. Let's see, you want a seating, window, aisle, nose cone, or do you want to be in the learning management system? But anyway, just kind of having some fun here. That's an actual thing for students. If you didn't get a boarding pass, we can get those out to you. I think we got a lot of those left.
And encourage them to save them and use them as bookmarks. They are kind of
Right.
big, but they still, it's a fun thing that they can remember throughout the year. So.
Right. It can become a collectible. Just have them come on for other missions. Right.
Thank you. I'm excited.
Awesome, Rosie. I
Hey,
can see your
someone just
excitement through
joined
the
us.
screen. Well, we're at the top of the
Let
hour.
me see if I can hold on. There, can you see? See, part of my problem is that I'm on my phone and I have to shut one eye to look at you all. And it's been a long week and it's only Monday. That's a little rough.
What day is it? Right?
But we're at the libraries, and Andy's library is going to be opening up, but my library is going to be staying curbside. So I'm setting up like a viewing platform for my ExoLab so that kids can walk up to a window and see it, and then try to engage them through social media. Plus we do curbside crafts and activity kits and STEM kits and things like that. So we'll be sending out, Andy put together, and his staff put together a little bean in a CD case. And we've got some other plant-based projects that we're sending out curbside to our students or patrons, I should say. I know they're students. When we're engaging in the back of minivans these days, and that's just how library service is right now. So, but we're really excited. We did the program last fall with our Michigan Library Association, the virtual panel, and that was really fun for me too. So I'm excited. I wish I were open and could share with the public, and so just trying to find ways to make it fun for everyone and get more people involved.
There you go. If you guys
So
want to, go ahead, Tony.
Are your students going to have their own account?
I don't know. It just at this point, not through my location. If Andy decides to do something, or we're going to be meeting on Wednesday to set up our experiments, and we'll talk more then.
Right.
But
I would encourage you to create an account for every student that are doing it with you, even at home, so that they can really log on and they can see and make their comparison and do their observation.
Well, we try, but when we're not able to go and actually do it for them, that makes it harder. So.
Right. Well, we have a digital open door here at Magnitude, so do not hesitate to reach out. And we're active on the weekends as well. So if you found you're at the end of the week and you still don't have a question answered, feel free to reach out to us. The hello is a general box, but Ted, Tony, or Lori will get to us directly. I think my number's out there as well. Feel free to text me if it's something you really want to make sure you get a quick response on. But we're just starting the mission, and this is when it gets exciting. We want to make sure you have all the tools available to you, and the website's working the way it should, and the experience is great for you and your students. So with that, I think we're on our way, right? So thanks for joining us, and we're really excited that you are. And I guess we're going to send notice out when we actually are getting our first live images. The installation of the lab, the astronaut floating around, putting it in, they record the installation, but it usually takes a couple of weeks before we get it. As soon as we get it, we'll make sure we publish it so you guys can share it with your schools, with your students. I think there's a certain level of excitement that goes with these rocket launches, and when you see the astronauts actually working with the project directly. So with that, I guess we'll let you have your evenings. Thanks for joining us.
Wait, I have one more question. Andy, are you going to have Greg Johnson coming by again?
We haven't talked with him recently, so I don't think so, probably. But last time he came, he was a great ambassador, so we were really grateful.
He is.
I think he's still down in Texas. He just comes up when the weather's good, right?
Right. It's not good right now.
Get to during the summer. Awesome. All right, guys. Well, have a wonderful
Okay.
evening and welcome to
Thank
space.
you.
All right. Be well.
Thank you. Bye-bye.